feel much honored and privileged to introduce Professor Mukherjee before you. Professor Mukherjee is a Masters of Pharmaceutical Science, PhD, and fellow of FIC and FICS, and is a professor in pharmaceutics and former head of the Department of Pharmaceutical Technology, Jadavpur University, Kolkata. He is a former DAD fellow Germany, and also he was a very important member of the our ADFI, Association of DAD Fellows and Invitees, care of Maximilian Bhavan. Professor Mukherjee can recall me very much. He's an ex-guest scientist, German Cancer Research Center, Heidelberg, Germany. He's also the coordinator, AICTE, Sponsored Quality Improvement Program, Nodal Cell Pharmacy, Department of Pharmaceutical Technology, Jadavpur University, Kolkata, and Joint Coordinator, Center for Advanced Research in Pharmaceutical Technology, Jadavpur University. He is also the convener and Animal Ethics Committee, Jadavpur University, and the former visiting fellow for the School of Pharmacy, University of London, UK, and a former Indo-Hungarian Education Exchange Fellow, National Research Institute for Radiobiology and Radio Hygiene, Budapest, Hungary. Is also a recipient of Finland government scholarship to work in university in Helsinki. He has worked as a guest scientist in German Cancer Research Center, Heidelberg, after receiving the Overseas Research Associateship Award, Department of Biotechnology, Government of India. He is also a former faculty of University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Works as an antigen technology and chemo prevention in cancer model. Also working on novel drug deliveries, particularly transdermal patches and nanosize, somal and neosomal formulations, as well as the nanoparticles. They have supervised many projects of different funding agencies, like UGC, CSIR, ICMR, DST, DBT, DRDO. There are many such things, and guided number of PhD scholars and master degree students. He has published his work in many international journals of repute, written a few books and book chapter, contributed very significantly as few patents in his process. He is the editorial board of some international journals of the repute, the recipient of UGC Research Award in 2009 and APTI Professor CEO Award in 2016 for research in pharmaceutical sciences. Received many other awards too, became the co-chairman of the Scientific Services Committee, LOC 56 Indian Pharmaceutical Congress in Kolkata. The first week of December 2004, he is Executive Secretary of Indian Association of the Pharmaceutical Scientists and Technologists, Kolkata, India. Delivered an invited lecture in the Presidential Symposium of 62nd Indian Pharmaceutical Congress at Manipal, Karnataka. So this is a very his short CV, and really we are very much eager to listen to him. I request Professor Mukherjee to start his deliberations. I think it is for half an hour timing. The time is 12:22. Maybe he should finish his talk by 12:50 or so. Professor Mukherjee, please.
So thank you very much for the exaggerated introduction. And what I like to know from you that whether the slide is properly visible to you all or not.
Yeah, yes, you are very much audible. You are also, the slides are very much visible. Yes.
Oh, thank you very much. Thank you very much. So just, 25 to 30 minutes time. So I do not like to waste any longer. Waste time any longer. I'd like to congratulate Dr. Chakraborty, Dewey Chakraborty, and also be grateful. She has invited me, and also the director of the institute and all other people who are associated to organize this program. So basically, you understand that therapeutic potential we are just going to discuss on the nano size drug delivery system. So few things are important. One is the therapeutic potential. Second is the targeted. Third is the nano size, and finally it's the delivery system. So why I just want to tell you why it is important. The targeting is very important. So like in a village, say a boy is just going beside the mango tree and a ripe mango is there. He has taken a stone and pelted he has spelled it to the mango. If the direction is correct, if the targeting is correct, he can enjoy the mango. So targeting is very, very important. And you know that most of the cases, whatever drug we are taking nowadays, it is non-targeted. Non-targeted is very, very harmful because you know that you understand the Avogadro's number, 1 gram mole contains 6.023 into 10 to the power 23 number of molecules, huge, huge, huge. If you write on the board, you cannot even read it, what is actually the number. So you know that when you are consuming the drug, even you are taking billions of molecules in your system, and these molecules are not, you know, your friend. So maybe if you just, if you want maybe 50%, 60% of them, and this remaining 40% will be harmful for you. This is very important. I just want to give you a simple example, you will understand. Initially, the paracetamol, dose of paracetamol was 500 milligrams. It was working perfectly. All on a sudden, I found that, because of no such, you know, authentic, you know, convincing approaches, it has been raised to 650 milligrams. So 150 milligrams you're consuming additionally, and you understand how many molecules you're consuming. So these molecules, this number of molecules, additional molecules, they are not your friend. And you see the adult dose is fixed. So all of the adults are not having the similar body weight, similar, you know, what you call is this body surface area, height. So the person who is having the body weight 45 kilogram and the person who is having the body weight 75 kilogram, both of them are consuming the same 650 milligram of the drugs, and this is absolutely injustice to the body. So you see this enormous number of additional molecules obviously doing some harm, and it is spreading all over your body. So it is important that if we just want to send the cargo to New York, and if we can bring the cargo to New York, exactly if we just drop it there, it is very nice. But if we just drop it on the way from here, Calcutta to New York, in New Delhi, in Germany, in London, and even in Atlantic, you see half of the cargo will reach there. The additional cargo is spreading in your body, and they are harming your cells, your health. So targeting, we understand, is very important. Secondly, we are working on the carrier system. We are not working with drug. We are carrying the drug. That means a person is standing beside a river, and we want to carry that person from one side of the river to the other. So this is the carrier system, but here the thing is this carrier system is nano size. Nano size, some advantages is there. So we were, all of you, I think at these days, during these days, we understand the importance of the nano size formula. We have the idea, but it is just, you can say, the of micro size. 1,000 nanometer is the one micron. So you see, even bacterial cell is much larger in this nano size, larger than the nano size. Virus is, normal virus is 40 to 100 nanometers. So we understand proteins, and you see there 4 to 10 nanometers.
Yeah, Professor Mukherjee. Yeah, Professor Mukherjee. Hello, Professor Mukherjee.
Hello.
May I request Professor Ketu to keep you in mute?
Can dissolve the products, and they have different colors also. So it is depending on the size also, physical properties changes. What we are going to do here, basically what we want to focus here, targeting. how we can target the drug into the cells. This is maybe those who are working in the area, it's not much, they are not much unaware of this thing. So here the drug has been put in the nanoparticle, in the nanoparticle, you can say this is a nanoparticle, and we are conjugating aptamer on the particle surface. So aptamer basically, they're the DNA, single-stranded DNA, and the deoxyribonucleic acid, and it's a fraction, small fraction, maybe up to 50 nanometer, even sometimes. I beg your pardon, 50 bases. Sometimes it may be less. And what is important, that this is a single-stranded DNA. When you will put it in the system, they will be destroyed. They will be destroyed because different DNA enzymes are there, even the RNA enzymes. So single-stranded DNA will be split, destroyed. So we have to protect it. How can we protect it? We cannot change the bases. Bases, if we change, it will be perfectly mismatched and it will not work. So we cannot change the sugar. It will change the character. So like deoxyribose sugar, we cannot change. So what we change is the phosphodiester linkage. Here you see the X is there. This X is, basically you are replacing this oxygen by sulfur or maybe another oxygen because the double bond oxygen was there. So you can just change it because then this DNA enzyme cannot recognize the sequence and it can enter, it can be stable inside the cell. So this particular work already published, we have recently published is molecular therapy nucleic acid, and I want to focus some part of this work because in this short time, it's not possible to give the entire part. Why I'm just giving you this reference, those who are interested or those who have some queries related to this, you can directly go into this manuscript and see the details of this particular study. This is the nanoparticles we have prepared. This is same, FESM, rather I should say, field emission scanning electron microscopy. This is, this 10 picture, these are the AFM, and the main focus is whether these nanoparticles are tagged with aptamer or not. So these are the nanoparticles. You see, these nanoparticles, since they're heavy, they were unable to move from the wells, and these are the different aptamers, free aptamers. It has moved, and they're the ladder, you understand. So basically, we have worked with 2 different aptamers. Actually, we worked with 5 different aptamers. Out of these 2, actually, seems to be better. And that is, we named it here L5 and L2, and we worked with them. What we have seen. So you see, we have taken initially 2 hepatocellular carcinoma cell, HepG2 and HUH7, was 7, HUH7 cells. So, and we have just given, the first initially we want to see whether these aptamer conjugated nanoparticles are entering into the cells at all or not in vitro. So you see, what we found that, yes, if you just see this particular figure, this is, facts. So flow cytometer, flow cytometric data. So here you see, this is what you find. This is aptamer L5 and others are there. This is control. So everywhere you see aptamer L5 have taken, much more amount inside the cell. So cell has taken up them much more. This is for 2 hours, 4 hours, 2 hours, 4 hours in 2 different cells. One is this, what you call HepG2 and the other is HUH7. And then IC50 value we have done, and you see these data are there. Here also you can just see the IC50 value in this L5 conjugated nanoparticles, where the drug is teclitaxel actually. So it has shown, the cytotoxicity, much more cytotoxic because it has taken up well by the cells, of course, in the different cell lines we have taken. And here you see this is percentage of inhibition. This is Chang liver also and WRL68. It is actually, these 2 cells are the normal cells. So if you want to study it, always you have to take the control cells. So we have taken 2 control and 2 carcinoma cells. Now these are the data of the control cells, uptake data. You see, not much difference is there in the L5 in 2 hours, 4 hours with respect to the control, not much difference. This is actually very encouraging. When you are just going for this drug delivery system, when you are going for this targeting, you know, the first thing is you have to say, you have to find out, at least you have to detect whether there is a comparable, comparatively more uptake is there in case of, you know, this targeted delivery in this targeted cell as compared to the normal cell. So it is there. We have achieved, we can see, it is clearly visible that in normal cells, this is basically what I mean to say, is WRL68 and Chang-Liver cells, though their uptake of these aptamer conjugated nanoparticles are more or less like control, so much less. Then what we did, we have analyzed, you know, the cell cycles, because it is going inside the cells, and whether it is able to release the drug and working in the proper way, because drug is already known, its mechanism is already known, and paclitaxel we are using. Paclitaxel basically, you know, blocks, inhibits, particularly the, you know, what you call is mitotic cell division. So here you see, in this case, if you go for this L5, you know, it is 55%. 55 is the data you can see as compared to the other phases, S-phase and G1 and GSG1 and G2GM. So G2GM is this particular phase where the actually prepared cells undergoing division, mitotic division. So here you see this is capable, you know, to function. So this is also another, you know, satisfactory data that yes, the drug is also releasing and killing the cells, because as you have got it in the what you called is this previous slide, you have got this data here, but this IC50 value, but still you have to confirm in the secondary, you know, what you call is the experiment. So next you see DNA fragmentation. What we did also, here you see the main important thing is in the DNA fragmentation. Once again, you see your L5 data is very, very different than others. It's a hugely different. So L5 data is hugely different, 86,000. And here in other cases, you know, that it is much less. We have also used the marketed formulation. We have also used this free drug. PF is the free drug, and also we have used other, you know, targeted formulation along with the aptamer, that is galactosamine and transferrin. We have used for this, but you know that they did not work comparatively significantly, so we rejected them, and we actually started with working this L2 and L5. So here, whatever I have said, and then our projection was whether it is going for the apoptosis or not. Because, you know, this is very important to understand this apoptosis, because the cell, if the drug kills the cells by necrotic process, this is not wanted, because this is not the process of killing the cells rightly. It will cause the toxicity in the tissue heavily. So we never prefer that in case of the drug delivery, we never prefer the drug should cause necrosis in the cells. Rather, we prefer drugs should cause, you know, apoptosis. So, the power disturbance is there. Number of times, frequently, the power is going and coming back. Anyway, so what I have seen here, you see, in case of the, we have used this acridine orange and ethidium bromide. So, you know, this normally in case of these cells, when it is having apoptosis, you will find the stomatic condensation is there, cells shrinkages are there, membrane blabbing is there, there is also nuclear fragmentation. So this phenomenon, also the formation of apoptotic body, so which is there. And I just want to tell you that in the case of apoptosis, normally in live cells, the green, normal green nucleus is there. In case of this apoptotic, early apoptotic cell, you will get the bright green type. Of course, here the perinuclear regions are there. Also, the formation of these apoptotic bodies and perinuclear chromatic condensation is there. And then also late apoptotic, you will find to orange to red. But in case of necrosis, you will also find red. But there you will not find the nuclear damages. There you will find the flat nucleus. And here you see this chromatic condensation and all these things you will find here. That indicates late apoptosis. But here you see this is more or less like live cells. No much change is there. These cells are actually necrotic cells. So you have to understand this difference. And we have found it is in the marketed formulation. And this is also L5 and L2. You see, they have lots of, what you call is the late apoptotic cell. Similar picture is there here, and you will find this late apoptotic cells. Here it is L5, and this is L2, and this is the marketed formulation, and the other two, this is pre-nanoparticle control. And this is for these two HG2 and HUH cell, just for the comparison side by side. Now you see, we have tried to determine the mitochondrial membrane potential by GC1 staining. GC1 basically, it is green, and when it goes inside the cell and forms the aggregate, it becomes red. And this red to green ratio, when it is enhanced, that indicates that there is a damage of the mitochondrial membrane. So what we found is here also you can see the data of course, this L5, and here it is, and here it is. Similarly, you see, that indicates here also, and here also, you see, there's a huge difference as compared to control and others. It's all here. You see, in this particular quadrant, and that is, the late apoptotic region. This particular quadrant is actually, the late apoptotic region. So the first quadrant, this is for the live cell. Second quadrant for the, this is in the, oh my God. So first quadrant is for the control cell. The first quadrant normal, then it is early apoptotic, late apoptotic, and finally you can say this is basically the necrotic cell. So you see here in this data, always here you see it is, you are getting more, this late apoptotic cell, here little early, here late apoptotic cell. So you see the findings are there. And in other case, this cases, basically this is related to the mitochondrial membrane potential, this P3 and P4 live, and then the apoptotic, live and apoptotic part is there. Now we have also determined the ROS. I can just quickly go for that. Once we are convinced that yes, it is actually causing apoptosis in the cancer cell, and it is also well taken up in the cancer cells, but in the normal cells, its impact is really not at all significant. So in that case, we go for the animal models, and in the animal models, we have taken some proofs, and then we have actually treated the animals with carcinogen, basically diethyl nitrosamine, one injection intraperitoneal, and followed by, we have given 2 acetylaminofluorine. So this is the model we have, and then you see it runs for 3 to 20 weeks, and then there's a gap, and then we have treated the animals basically with this aptamers and these formulations, and then there is a gap again. You can say pause after the treatment. There is 36 and 38 weeks, 2 weeks after that. We dissected the animals, and we have just studied the histopathology and some enzymology, also some molecular markers. So histopathology, this is, you see, what we have found basically in the carcinogen treated animal, carcinogen control, basically what I want to mean. Lots of neoplastic cells are there in the first case, hematoxylin, eosin hematoxylin. You see, there you will find that basically this is a ground glass type of focal lesion. Second case is mostly basophilic, and third one is a very interesting type of focal lesion. This is you can say the precursor type of hepatocellular carcinoma lesion. These are basically these cells. They have pointed nucleus, maybe sometimes more than one nucleus, and basically they remain in the peripheral area. So they looks like empty, void, but they're not void. They're cells, and they are actually spongioblast type of cells. This hepatitis, it is called basically. And then after the treatment, we also try to find out the changes at the different stage. We have taken the histopathological slides from the animals and see the improvement. Really, it is interesting. So you see, favorable in case of the first slide. You see the carcinogen treated carcinoma, when it is big or lots of necrotic cells are there, and eventual improvement you can see also in the other slides. After the treatment, these are the quantitative data. So this is available since in the published material. I'm not going to discuss about this data. This is basically, we have quantified these focal lesions of different sizes, and you can see these normally carcinogen treated animals treated with this L5 and L2, they have much less, the smaller lesion, the larger lesion compared to the smaller lesion. So that means it is delaying the progress of the development of hepatocellular carcinoma. The markers, also the other marker proteins we have selected and quantified, basically, this apoptotic marker, and also some anti-apoptotic. You can see this thing. This is also there in the journals. So because of the sort of time, I'm not going to discuss all these things. Basically, what I want to focus here, that these cells are getting tired by means of the apoptosis. Favorable apoptosis is seen quite in the protein markers also. Then we done for pharmacokinetics, and you see the formulation nanoparticles L5, L2 there, obviously, and the free drug and the commercial formulation, you see, they are sustaining more in liver. So this is also important in terms of the in the point of view of this formulation. This is for this normal cells treated with L5 and L2. You see, no much changes there. This like normal histopathology, and finally, you see, this is overall biochemical parameters we have shown here. SGOT, SGPT, and alkaline phosphatase. These are basically, the liver enzymes. This is related to understand the condition of this liver. And here also, you see, no much changes of this liver parameters, liver enzymes is seen in case of SGOT, SGPT, as well as alkaline phosphatase compared to the normal cells in the normal treated animals. That means it is not, this formulation not creating, what you call this hepatic damage as such. And then this formulation, we have found out of these two formulation, of course, L5 type is very, very potential. And then we wanted to know that where it is actually getting bound, how it is entering into the system. We went for the formulation L2 and L5 through this homology model, and we found that this is done by my colleagues. Of course, I am not expert in the field, and we found that it is actually targeting histone protein 70 and TAC 72. These two proteins are possibly the location of binding this aptamer to enter inside the cancerous cells, because these are found mostly in a much high expression. You will get this particular type of, hits of protein and TAC2, SAC72 in cancerous cells. My last small presentation, this is related to very interesting thing. We wanted to send the drug into the brain because it has to cross the blood-brain barrier. Docetaxel is the drug, and we developed this nano formulation here. This is, basically the nanoliposome. Nanoliposome, advantage of nanoliposome is they are mostly very, very small. You can get it even 30, 40 nanometer size, and they're mostly uniform. So this nanoliposome, once you prepare it, this is the size we have mentioned here. After the in a liquid suspension, so obviously it will enhance the size a little bit. But anyway, you see it is less than 100 nanometer. You can understand. So this is the cryotem, and cryotem is very important to understand liposome, because liposome it's not a solid structure like marble. It is a what you call it's a flexible structure like balloon. So you have to understand its membrane is very important, and you see this is unilamellar mostly, but there are in this nano range. Normally, people say multilamellars are not found, but although it is less, but there are few multilamellar liposomes are found also here, and this is unilamellar. So you know that at that nano range also, all the liposomes are well protected. That means membranes are not damaged. Then we actually mixed FITC, and we wanted to see whether it is reaching in the brain. Of course, we have found that they're reaching in the brain with the confocal microscopy. And then this slide is very important. You see, when we use this, this is normal. This is the normal formulation. We didn't put it in pre-drug. You can say it is not reaching at all in the brain. When we put it in the normal liposome, this phospholipid and cholesterol, it is reaching in the brain. There is no doubt it is reaching in the brain. But when you see we have put it in what you call this this particular chemical, I will discuss, and that chemical is able to bring it to the brain in an extensive way, much more higher in amount. And that is a target, and thereby you can reach it into the brain. What chemicals we have used? This is the chemicals, 1,2-diacetyl glycerol phosphatidylamine. This is DSPE. This is the chemical which is there in brain. So when the drug is not reaching, formulation is reaching, the nanoliposome is reaching, and it is going to the brain, but not that desired level. What we thought that let us incorporate certain amount of, this brain lipid, and let us see whether it is going to the brain or not. We found, yes, it is going to the brain, and it is really, we have achieved it, and that this is a projection that you can also target the drug into different areas, not only by direct targeting approach, by this indirect targeting approach. This is the blood level and brain level of this formulation. You can see this distinctly different when you are putting it into this nanoliposomal formulation. Thank you very much. I hope I didn't take more than 5 minutes, and thank you all. However, if you have any question or queries, do not hesitate to ask me. Thank you.
Thank you, Professor Mukherjee. Thank you so much. Yeah, ultimately, you have been able to restore it and showed the remaining part of your presentation to us. It was really fantastic presentation and quite informative and excellent as well. Because of the acute shortage in time management, we are not throwing the questions. The questions are in mind amongst us, and whenever we will meet you or contact you, and then those questions will be clarified later on when it will be placed to you. So we on behalf of
one thing I just want to interrupt you for a moment. If anybody is having any question, and because of the time constraint, if it is not asked, do not hesitate to email me, because my email is available in the net also in the university website as well as maybe in all of these publications. So do not hesitate to email me. I will be glad to answer because just presentation is a part, but I believe until and unless people clears it down, it is not a complete presentation. So anyway, if anybody has any question in mind, do not hesitate. Please contact me by email. I will be proud to. I'll
Sir,
be rather happy.
a small question is, it
Yes,
was very astounding to know that the normal healthy cells are not getting affected, and with your formulation, and to the extent the apoptosis is occurring with the formulations which have been designed by your group. It was very astounding actually, and I wish to have the mechanism, but maybe due to time constraint, it will be difficult right now to.
I just in a small time, I will take small time to answer this question. Basically, you know that this hit-shock protein and this TAX72, we believe that it is able to internalize this formulation into this cancer-specific cells, which is not there in case of these normal cells in that way. So it is reaching much more in the cancer cells in the short period of time and introduce apoptosis, thereby, you know, it is just killing the cancer cells. So this could be the possible mechanism or proposed mechanism because apoptotic data says like that.
Is the protein specific? That's what your
Yeah, protein specific. Protein specific. Yeah.
Protein specific. So, sir, can you please make your video on so that we can have a small snap?
Yeah.